Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
All right, so I am very pleased to introduce to you Crystal and Matej. We're both fifth-year grad students at Berkeley, although apparently this does not mean that they're equidistant from graduating, because Matej is finishing next year and Crystal just told me too. But they have spent quite a bit of time working with a bunch of people, including me, on gene sequencing. And I can't speak for them, but for me, this is one of the most fun things I've ever done. It's just amazingly cool to learn that knowledge about hash tables translates into biology. So, so they're going to get to tell you about the project that we've worked on, and they're going to team speak. I don't know what order they're in.
Yeah. All right, so I'm Matej. I'll start it off. So this is a project that a bunch of people are interested in now. A lot of the people you see up here are from the AMP Lab at UC Berkeley, which is a new lab that started a year ago that's focused on big data and large-scale machine learning and data processing. And we're also working with Bill and Ravi from Microsoft and with some folks from UCSF, including Taylor Sittler, who's here today. He's a medical researcher there. So yeah, let me just jump into this, why we're doing this. So I think if you've seen some biology in the past, you'll know DNA is the molecule and the code that orchestrates all the activity of a living cell. And because of this, DNA is a central element in a lot of diseases. It either has, there are differences in the DNA that cause the disease, or it's like actually involved in propagating it somehow. So in particular, what DNA does is it encodes how to build these worker molecules called proteins in the cell, and also signaling information about essentially when to build them. So everything the cell does is guided by that. And two ways in which it ties to diseases is, first of all, in cancer. Cancer is essentially caused by the DNA in some cells accumulating enough mutations that the regulatory mechanisms break down, and those cells start multiplying without the right regulation, and just start consuming all the resources of the body. And on top of that, there are hereditary diseases, which are just if there's some mutation that's passed down that causes a problem. DNA also affects susceptibility to various drugs, and it's an important thing to understand what's going on in the cell. And one of the things that's happened in the past decade is that the cost of actually reading the DNA from a cell has fallen dramatically. And it's fallen enough that it's starting to actually be used in clinical medicine for treatment. So here's a picture of that, and this compares it against Moore's law. This is the cost of sequencing one human genome. And if you go back to around 99 and 2000, the Human Genome Project cost several $1 billion to sequence the first human genome, and that was a great accomplishment. If you fast forward a bit, this year you can actually get your genome sequenced for about $3,000. And next year, a couple of companies have announced that they will do it for $1,000. So this sort of magical $1,000 genome is really on the horizon. And I think if you work out the rate, the cost has been falling by something like a factor of 3 or 4 per year, and it's actually quite a bit faster than Moore's law. So we're able to actually read this stuff now. And being able to read this stuff can impact a lot of areas of medicine and, of course, of biological research as well. So here are just some examples. So in cancer, the most exciting thing about this is cancer is typically caused by a bunch of mutations that affect many different gene pathways. Like these are groups of genes that work together to perform a function in the cell. And often cancers that look very different, like a skin cancer and a lung cancer, might involve the same pathway. And by actually sequencing it, you'd be able to figure out which pathway is involved, and also which drugs you should use that target that particular pathway. So there are many targeted cancer drugs now that will work really well if one of the possible things is going wrong, and will have no effect if something else is happening. For infectious disease, like just common viruses and bacteria, one of the nice things you can do with sequencing is you can really quickly find out which pathogen a patient is infected with. You don't have to do these sort of crazy diagnostic tests where you look at the symptoms. You can actually get some DNA and see what's going on. And another thing this has been used for is identifying new diseases such as the H1N1 flu by actually sequencing and saying, hey, this looks different from stuff that we've seen before. And finally, you can personalize medicine to individual's genome, so you can identify things they're susceptible to in advance. So for example, some drug allergies or side effects only happen to people with specific genes. You can know which inheritable diseases they have, and you can estimate the risk of different things going on. And there are some examples of this happening already. Just a couple of weeks ago, there was a series of New York Times articles about sequencing-driven personalized treatment for cancer. So this was a case at Washington University where they sequenced the cancer tumor that actually one of the researchers they had, and they figured out that it was actually susceptible to a drug that had been designed for a completely different type of cancer, and they applied it, and actually they managed to really induce a remission in that patient. There's another one that was, this was a case where, in the end, it didn't work out. The remission was only for a short amount of time. But similarly, they were able to use a drug from one cancer to treat a different one that it normally wouldn't be prescribed for. And there are some examples in sort of non-cancer things as well. So for example, diagnosing metabolic diseases, which is usually very hard. They can sometimes be due to mutations in mitochondrial DNA. And here they were able to just sequence mitochondria and figure out some of these diseases. Okay, so this is all great in terms of affecting medicine, but we're here talking at a computer research lab. So what is the computational challenge? So it turns out that although the cost of the wet lab part of sequencing is dropping dramatically, you have to do a lot of processing to actually put this data to use, both to understand what is happening, like actually how to put together the sequence for one human, and of course beyond that to understand which genes affect which diseases. We're not even going to get into that in this talk, but that's also a very hard kind of machine learning problem. But the reason that the processing is difficult is that the way the sequencing machines work is essentially by using massive parallelism, by reading many small sort of random substrings of the genome in parallel, and you get together these little random strings, kind of like puzzle pieces, and you have to put them together to see the whole picture of what was actually going on in that person's genome. And I'll show what that looks like and what are the steps you need to do that. So the bottom line is that the current pipelines for doing this take multiple days for each genome, and cost, if you actually work out the compute time, it would cost on something like Amazon or Azure, or to some extent, even if you buy your own machines, it can cost in the thousands of dollars just for one genome. So this is starting to exceed the cost of the actual sequencing itself. And yeah, question?
So the graph you were showing was just the chemistry part?
Yeah, just the chemistry part, exactly. Yeah, and the other, so this is, it's not great for clinical use, but it's especially bad if you want to scale up the amount of genomes you sequence for research and really understand these diseases, because people have to pay that many times over to sequence, 100 cancer patients and see what's going on. So the goal of our team is to build a faster and more scalable and more accurate pipeline that does this genome reconstruction, and that can be used both in actual medicine and in research, where you get a lot of different genomes and you want to compare them, or you just want to do this kind of computation at scale. And we have a bunch of people involved from a bunch of different areas. So we have a lot of systems people. This started out essentially with a bunch of systems people talking with Taylor and figuring out that we can do some of these steps faster. And Bill and Harvey are from Microsoft. We have Taylor and Arun from UCSF. And we have some folks in machine learning and in computational biology and theory that are helping out as well at Berkeley. All right. So what will we have in this talk? So I'm going to start with just an overview of how the sequencing process works and what are the computational problems. And then we'll mostly talk about the first processing step, which is sequence alignment, and also turns out to be the most expensive in terms of CPU time. And we'll talk about a new algorithm we've developed called SNAP that cuts down the cost of this step by a factor of 10 to 100 compared to the existing tools. So this is something that takes a problem that was essentially CPU bound initially and took more than a day for a human genome, and it turns it into a problem that's now basically I/O bound, and we can do it in an hour and a half. Then we'll talk about some ideas we have for further improving alignment, especially the accuracy, by taking advantage of the structure of the genome. And finally, we'll also talk about some downstream processing steps beyond alignment. So hopefully this will give you a taste of like the different types of problems that exist. Okay. Yeah. So let me just show real quickly with some pictures how the actual sequences work and what you have to do with the data. So the DNA itself that you have is a long molecule. Well, it's actually a bunch of long molecules, but in total it's 3 billion of these letters or bases long that encode the sequence. So you have a sequence of these 3 billion things. And the first thing you do is you replicate it. So you have a bunch of copies, or you just read a bunch of copies from a cell. And next, to actually sequence it, everything is done in parallel. So you split it randomly into fragments. You can just do it by like heating it up or something like that. And you get these little fragments of DNA. And after that, you can read the fragments in parallel. There are machines that do this by doing some clever chemistry to be able to read the sequence off of each of them. And the way it works is they kind of like attach it to a little thing on a plate, and they float around the complementary bases to the ones that are part of your DNA, and they read what actually sticks to it and in what order. And as they do this, they end up reading the sequence. So these little fragments are called reads, and they're about 100 bases or letters long today. But the whole thing, as I said, is 3 billion. And in a typical human genome sequencing run, you're going to get 10 to the 9 of these reads, which is basically covering each location in the genome about 30 times, and they each are 100 bases long. So now, how do you actually put these things back together? You can view it as a puzzle. So you just get this sequence of reads. And one way to treat it as a puzzle is, like, let's see what the picture on the top of the puzzle box is. You can use a reference genome, which is the genome that was sequenced back in '99 and 2000 and has been refined since then, where people use longer read technology and know how everything ties together. And when you look at the reads from the person, they're going to have some differences from the reference genome, of course, because it's a different person. And they will also have differences because of errors in the sequencing itself. But what you can do is you can take each read and see where in the reference genome it matches the sequence best. And then just place it at that location, like placing the piece of the puzzle on the sheet in front of you. And so this step is called sequence alignment. This is the one that we'll talk a bunch about. And what this lets you do is, once you've aligned all of the reads at each location, you'll get something like this, where a bunch of them align together. And they might all show some difference from the reference genome, or you might have some of them show a difference, some of them show noise because there is some error in the actual reading process. But then you can do kind of a voting algorithm to actually figure out what the base at that location was. This is really simplified. It turns out there's a lot of stuff that makes this harder. So this step is called variant calling. And we'll talk about it a little bit later. And just to give you a sense of the rate of differences between these, two people only differ in about 1 in 1,000 bases from each other. So that's pretty small. But the sequencing machines can have error rates of up to a few %, although it depends. Turns out some of the errors are also pretty biased. So this is kind of the error rate you're looking at. OK. So let's jump into the first step of this, which is alignment, and what we ended up doing for that. So the alignment problem, as I said, is given one of these reads and the reference genome, which is a big string, find the position in there that minimizes the edit distance to the genome. And again, to give you a sense, the genome is 3 billion bases, and the reads are about 100. Okay, so if you look at the current status, alignment is today the most expensive step of processing, and it's also important because you really have to map the reads to the right location to do anything downstream of that. So depending on the accuracy of the tools you use, it can take a few hundred to a few thousand CPU hours, and if you work out the math, it can take sort of hundreds to $1,000 of compute time. And the issue there is also that the faster aligners lose accuracy, so they typically don't align as many of the reads, and they support fewer differences inside each read. And the problem with that is if you have a place in the genome where the person really has like 5 differences in a row, and your aligner doesn't support that, you're going to systematically miss all the reads that map there, and you're not going to see that difference in the downstream analysis. So what we built, SNAP, the Scalable Nucleotide Alignment Program, is a tool that's 10 to 100 times faster than the current ones, and at the same time it improves the accuracy. So it has higher accuracy, and it also has a richer error model that allows for more types of differences. And basically you can give it a parameter k, the number of edits you allow from the read to the reference genome, and it will find a location with the best location with at most k edits. And as a result, as I was saying, we cut down this step from about 1.5 days to 1.5 hours. And this is done while reducing the numbers of errors in half on sort of real human data. Okay. So what do current aligners do, and how do we do this better? So there are two methods to do alignment. One of the earliest ones, if you've heard of BLAST, this is what it does, is based on seeds. So the idea here is you have the genome, you index just short substrings of it, of say 10 characters, and what you do is then you take your read and you take every 10 characters in here and look for an exact match in the index. So, say here we have just 4. Say these were the first 4 of them. This is where it matches. So you get some candidate locations to try, and then you place the read at each one and compute the edit distance, and you end up picking the best one at the end. Okay. So this is the seed-based method. And of course, you don't know, like maybe the first 10 bases match somewhere, but actually that's just by chance. Maybe there were some errors in there. So you have to try multiple seeds to really find the best location. And so you keep doing this for continuous, continuing seeds. This is one method. The other methods people do, actually many of the faster tools today, use a Burroughs-Wheeler transform to encode kind of a prefix tree of the genome, of like all the substrings in the genome, and then they search through this tree using backtracking. So this is, you're just going down a tree and trying to insert edits at different locations. But I'm not going to talk much more about this because it's a bit more complicated to explain. So what we do in SNAP is we've actually taken the seed-based method, but we've done a bunch of algorithmic changes and also sort of systems changes that reduce the cost of the most expensive check, the most expensive step, which turns out to be the local edit distance checks. So we leverage, on the one hand, we leverage just improving resources. One of these is the actual read length. So the read lengths used to be about 25 bases, and now they've gotten longer to about 150. Using this, it turns out you can really change the form of hash index you have and do quite a bit better. We also leverage higher memory. So our algorithm is designed for servers with about 50 gigabytes of memory. So we can use that. And on the algorithm side, we have a way to prune the search to reject most of the local alignment locations without fully computing the score. And this turns out to save a lot of time as well. So let me just explain the first part. So we're going to use these seeds to match a hash table of just exact matches in the reference genome. But we've chosen to use longer seeds than a lot of the previous aligners. And the reasoning for this is that there's a trade-off in general between the seed size and the probability of actually finding a seed in the read that matches the genome and the amount of false positive hits you have. Yeah, so if you have, so the human genome is about 4 to the 16 bases, and if you have a seed of 10 bases, and if you have a 2% sequencing error, it turns out there's a 19% chance that your seed that your seed contains an error. Otherwise, 80% of the time it will match. But also, you'll have 4 to the 6 or 4,000 matches just by chance, against the whole genome, just on average. So this is what people used in the past, and it made sense when they had very short reads. You couldn't take much longer seeds than that and expect it to match. If you go up to a 20-base seed, you have a higher chance of an error, but you also have almost 0 chance of it matching just randomly at a particular location. So at least if the genome is a random string, you expect to test a lot fewer candidate locations. And the reason this makes sense for a longer read is because the read is longer, you can take many more independent seeds to try. Yeah?
Are you making any assumptions about high repeat regions?
Right. Yeah. This is very simplified. So actually, high repeat regions mess this up. So some regions are, some seeds are much more common than others, and just big regions are replicated. Yeah. So you still have to search a lot. You have to search more than one hit for each seed usually. Yeah. We'll actually talk about that at the end. That's one of the things we're looking at. Yeah. Cool. Yeah, so just to show this, say you have this short read of 25 bases, you have an error there. Well, if you take a 20-base seed, there's pretty much no place you can put it without touching that error. But if you have a long read of 100 bases, and there are a bunch of errors, even with a 2% error rate, you do expect some seeds to work well. And so you can actually find matches for this in the index. So this is, like just an observation with some math of something we can do. Yeah.
So what technology allowed them to go from 25 to 100, and is that going to cause them to go to 1,000?
Right, yeah. Yeah, definitely. Read lengths are improving, and basically what it is I showed that picture at the bottom where like you attach one end of the DNA string and then you float these molecules around, and there's actually like they're fluorescent and there's a camera pointed at it that see which one is attached. And what happened is they've been able to attach more of them before they have to stop. So like after about 25 of them, there was too much noise to be able to attach more and like see which one actually got put there. And they've improved both the chemistry of like how those things float around and the camera technology to be able to do more. Yeah.
They're also moving away from camera technology as well, right? So they're using electronic reads and the bio-nano core.
Yeah, there are new.
There's other technologies that are more electronic reads, so you have way less complications. So we will see longer reads coming in the not too distant future.
I just was wondering, what is the order of magnitude we can expect in, say,
I think 10,000 is a...
10,000?
There are other people working on megabase reads.
Yeah,
that's farther out still.
It has a ways to go, but it'll be interesting.
Yeah, the 10,000 base pair they seem to be doable. There are sort of prototypes that are out there.
Yeah. It turns out backtracking is a bad strategy for length 10,000 with higher.
Yeah, right. Even for 100, we'll see. Yeah. Yep. Yeah, it's a good question. Yeah, so these things are getting longer. Yeah. Okay, the other thing we do is very simple on the index side, but it actually helps a lot, is a lot of the existing tools were designed at a time when server memories were a lot lower. And for example, if you index 10 base pair seeds, they only took the non-overlapping ones, like the one, position 0 to 9 and then 10 to 19, whatever, all these disjoint seeds. What we do is we just do a sliding window and index every substring of that length. And if you think about it, this means our index has 10 times or actually 20 times more seeds in it because we're using 20 base pair seeds. But it turns out that if you pack the bytes nicely into a hash table, you can fit that in a very reasonably sized memory. So we are able to do this with 39 gigabytes of memory for the human genome. And this is important because looking for a seed and not finding it in the hash table is actually really expensive. It's like hundreds of cycles because it's an L3 cache miss. Because even with a small hash table that they had before, it's not going to fit in your processor cache. So it really helps to actually have this. But the other part, so the algorithmic part that's really different beyond this is the way we do the local alignment check. As we talked, as I mentioned before, it turns out that the genome is not a random string. There are a lot of areas that are quite similar to each other. So for many seeds, you'll find them in a lot of locations. And in general, for many reads, you'll have to test a bunch of candidates before you find the best one. And this is where all the cycles in the algorithm go, essentially. Like at least 90% are going into this. So what is our insight here? So the thing we figured out is that at the end of the day, to actually map the read, you only care about the best and second base locations where it aligns. So if the place where it aligns best, say it has edit distance 1, and the second best place it has edit distance 5, you can be pretty sure that it came from that best one, and you're just going to align it there. If the best is at distance 1 and the second best is at distance 2, then maybe you're not sure, and you're just going to tell the downstream analysis, OK, I'm not sure where this read goes. You might give it both locations or something like that. But these are kind of, to confidently call a read, you just need to know the best and second best locations. So how can we use this? We place the traditional edit distance algorithm, which always computes the full edit distance at each location and is a quadratic time algorithm, with one way you can give it a limit on the edit distance, and you can tell it if the distance is bigger than this, I don't care how big it is. Just stop early and tell me it's bigger. And so we have this algorithm. The complexity is only n times the distance limit. And we lower the limit as we find more hits and improve the best and second best locations. Yeah?
So is n the larger of the two strings?
Yeah, n is, it is the larger, except here we're only comparing locally at each place. So n is like 100 bases. It's the length of the read. It's just we mapped it to one place with a seed. We take those 100 and the 100 that we have. Yeah. So with this algorithm, you can lower the limit, and you can also arrange things so that the first hit you find actually has a low edit distance and start out with a low one.
Isn't the Yukon basically a banded edit distance?
It's a little bit. So it's kind of like filling in only the diagonal of the matrix, but it's actually it's a bit nicer than that because it only uses like order D space. So it only like tracks how far you can go down each diagonal. So that's actually kind of cool because it fits in the L1 cache of the processor as well. Yeah. But that's what it is. Yeah. OK. So here's like the actual algorithm. I'll just step through this to show the way we actually do the pruning. So basically what we do when we start the D limit to be the maximum distance plus this confidence threshold C, which is like how far away we want the best and second best read to be to call it as unambiguous. And we go out and extract seeds from it. And for each seed, so this is the confidence threshold. Yeah. For each seed, we find the locations where it matches in the genome. We actually prune seeds that have too many locations because some things are just too repetitive. We look for a better seed that doesn't have that property. And we add candidates to a list. Once we've tested a minimum number of seeds, we look at the candidates that matches the most of them. And the idea here is we're going to do an expensive edit distance computation. Let's find the candidate that will give us a low D limit for the next one. So we score that candidate. And the next thing we do is we update the distance limit. So there are two cases for this one. So we look at the best and second best hit. We keep tracking them as we go along. And there's two possibilities. So if the best is much better than the second best, and we have this confidence threshold C, then we only care about finding other hits within best plus C. Because the idea is if there are hits with a bigger edit distance than this, it's not going to change our result. But if we find one in this window of up to C more, we're going to say maybe we're not confident about the match. So that's the first case there. Second case is if the best and second are already within distance c, then there's no way that finding guys bigger than the best is going to help us. The only thing that will help us is if we find something much better than the best that's up in this window here, and there's nothing between it and best, so we can be confident about it. So we only search up to best minus 1 in this case. Yes, so that's what we do with that. And the final thing is there's a trick you can do to stop early. So if say you found, the best is in distance 2 and maybe you only care up to things up to distance 4 beyond that, what you can do is if you've drawn at least 5 disjoint seeds from the read, you can actually stop right there. Because if you've tested a bunch of disjoint seeds, you know that any read you haven't yet put in your candidate list, so it didn't match any of the seeds, has at least that many errors in it. So if you've tested 5 seeds and there's a location that you haven't yet found through the exact matches on those seeds, then it must have at least 1 error in each of the seeds, so it must be at least distance 5. So you can actually stop early over here. So this lets us stop the search as well. Okay. So just in terms of the results, here are some numbers comparing SNAP to 2 of the commonly used aligners today, BWA and SOAP. They're both actually based on the Burroughs-Wheeler approach. And we can see with the existing aligners, so we're showing 3 numbers here. This is on simulated data, which lets us know where each read actually came from. And we're showing the percentage we aligned, the error rate, and the speed in reads per second. And here you can see BWA and SOAP give you a trade-off between accuracy and speed. SOAP is a little faster but makes a lot of errors. And SNAP actually matches the percent aligned beats the percent aligned of BWA. It has half the error rate, and it's also going about 30 times faster. Okay.
And that's on 1 core. We also scale better.
Yeah, that is true. Yeah, yeah. Okay, so this was 100 base reads with 2% differences. This is kind of the current reads you have today. Another cool thing, so in SNAP, you can actually tune some of the parameters, the max hits you check for each seed, and trade off between the accuracy and the speed. So if the speed, if that's too fast for you and you are okay waiting for a day to align this stuff, then you can tune SNAP to give you higher accuracy. So we show you could align with only 0.01% error if you go about 3 times slower. Or if you are okay with the error rate of BWA, you can tune it to get higher speed. So you can trade off between these. Yeah.
How do I read the percentage aligned? So the remaining 8 to 9% are the errors of the algorithm?
They are, no, they're just reads we couldn't align. So the main reason why we couldn't align them is, so these are the ones we confidently returned 1 location for. For the other ones, there were multiple locations where they matched well. There's also usually like maybe like 0.5% where they have too many differences and we don't find any location for them. Yeah. But this is how many we're confident about. Like how, yeah. Yeah.
So if we had a perfect algorithm, that would be 100% alignment with 0 errors.
No, it would actually be only like, I think in this case it would only be like 93% or something like that, because you can't, oh, 94, okay. Yeah, because some regions are exactly identical, and you can't know where that came. So we're not counting those. But it would have 0 error, yeah. Yeah, and actually even the error, because of the model, you might have 2 errors that put you closer to something than where you actually came from. But I think you could calculate what that is.
About half of the errors pick off where that. So the idea is, if you get either a mutation or an error in the read by the machine, it may move the string from the place that it came from to actually be closer in edit distance space to the wrong location, and then it's impossible to do this. I mean, you might be able to just say I'm not going to call it because it's too close to too many of them. We actually did that some, but
yeah, yeah. Okay, so this is kind of with today's data. One other neat thing is that if you have reads with a lot more errors, which some of the future technologies will have, and also which will happen if the person has a lot of mutations in one place, SNAP still performs pretty well. So here with 10% error, the existing aligners kind of fall over. They align less than 20% of the reads. And they actually go a little faster because it's easy when you're not aligning anything to go fast. But we still do okay. Yeah.
So it would be nice if you could combine this into just one number % error. You would need to incorporate the downstream processing.
You would do, yeah.
So what happens when you take just an off-the-shelf downstream processor and
right. So if you compare SNAP with others? That's a really good question, yeah. We don't actually have a complete answer for that yet. We've looked at does misalignment affect downstream callers, and we've found places where it does, like where BWAs misalign some reads consistently, and you call mutations that aren't actually there. But I don't have a good sense yet. Part of the issue is there are other things that are hard in the downstream caller that you have to like get right first before this makes a huge difference. Yeah. But we think it will help eventually. Yeah. Yeah. Okay. Yeah.
I mean, I guess to frame it somewhat, there's in most of the current processing pipelines, there's a second step of low degree alignment that's done after the initial alignment.
I didn't actually plug this
in. That's because I guess about 30 to 35% of the reads they expect don't map to the right location.
30 to 35%.
Yeah. This is now looking at cancer genomes, which are a little bit more complicated than normal genomes.
Yeah. In cancer,
there's significant room for improvement.
In cancer, also a lot of the DNA, like replication and sort of checking mechanisms break down, so you get a lot more mutations. Yeah, like weird things happen. Yeah. Okay. Yeah. And let me see what else I wanted to show. Oh yeah. So this is another thing we wanted to show here is as reads get longer, which they will in the future, SNAP actually scales better as well. So the existing tools, the fastest ones today have this backtracking, which is a bad idea when the reads are longer because you have to, backtrack at more locations. Whereas SNAP actually does better because you can use longer seeds and you can get more disjoint seeds and filter out a lot of the locations just by number of seeds matching. Okay, and we also did some analysis of the Speedup. So one of the nice things is this heuristic where at the front we test the read with the most exact matches of seeds. Means that usually the first candidate we score is actually the best one we'll find, and we get a low delimit at the start. And we eliminate 94% of the locations without actually scoring them. And 40% are just because the number of seeds didn't match. So this cuts down on the time. And the adaptive edit distance threshold also helped by about a factor of 4. Okay. And finally, just to wrap up this part, we've been doing a bunch of stuff after that, I don't have too much time to go into. So one of the things is we've generalized the algorithm to work with what are called paired end reads. This is when, this is actually the most common type of read. You get a bigger molecule, like 500 bases, and the machine reads 100 bases off one end and 100 off the other. And it can't go all the way into the middle. So now you get to align kind of two strings in a place, and you know some constraint on how far they can be from each other. And there's a bit, you can use similar ideas in this problem to actually align them as well. As Bill was saying, we've spent a bunch of time into making this scale well, and it does. This is on up to 32 cores. And part of it is because we're careful about how many memory accesses and cache misses we do. We're careful to prefetch data, and basically like having systems people look at this actually does help improve the speed to the point where it matters to practitioners. So that's kind of cool. And we've also run it on some real data. This is numbers I showed before. So these are real reads. Interesting thing with real data is a lot fewer of them align because there's some contamination and just like stranger things happening in them. But in this one, we're able to, again, sort of mirror the results in the simulation and get higher accuracy and also go about 20 times faster. OK, so that's the part of the talk that I had. Crystal's going to talk about what we're doing next. Yeah.
Would you consider this a solved problem? I mean, given the low error rates and the speeds that you're saying you're at IO bound,
right?
Are you guys done?
Right, it's a good question. I think the accuracy can be improved even further, and we want to explore this more to see when it matters. But especially for things like cancer, there will be more mutations, and also there will be things like pieces of the chromosome that usually aren't together get cut and pasted next to each other, and you might want to detect that. So now it's like when half the read aligns in one place and half in another. In terms of speed, I think the speed is pretty good, although there's always like, this is one genome in 1 hour, but there are people who have like 1,000 genomes data set. If they want to realign that using SNAP or using higher accuracy, that might take a while. Yeah, but so we'll talk a bit about like what we're doing for accuracy, but we also want to look more at the downstream steps next because there's more unknowns in there. Yeah. Cool.
Okay. So yeah, as Matej mentioned, we're kind of capping out on speed, but the accuracy is actually something that we still want to look at more because so as far as alignment goes, there are two parts of the process, and one is finding the good candidates to check the read against, and the other is quickly checking against all the candidates so you can find the best match. So in the first case, we've pretty much narrowed that down, and we're getting a good list of candidates, but in the second one, we still want to make some improvements there. So what we've noticed in doing a sensitivity analysis to what parameters tend to affect the performance of SNAP is that really the only parameter that makes a big difference is this max hits. And so as Matej mentioned, this is the kind of the cutoff for when you have a seed, how many hits it has in the table as to whether you'll consider it or not. And so what we noticed when we varied that max hits parameter is that the error rate does go way down as you get a higher max hits, and you can also align a higher percentage of reads. So what this means is that at first cut, you would think that a seed that matches in a bunch of places is just indicating that your read is just matching to, it's going to be ambiguous because it's matching so many places. But what we actually find when we are willing to consider more and more places is that for some of those reads, we actually can find an unambiguous best match in the genome. And the reason this happens this way is because, as you were kind of alluding to in your question, the genome is not a random string. It's actually highly redundant. And what makes it difficult as well is that it's not exact duplication that's really the main factor. It's similar duplication. So this is something that we really want to be able to consider more hits per seed. But then, of course, the downside is if we do consider more hits per seed, the speed takes a big hit. So how can we improve our accuracy, reduce our error, get a higher percent aligned while avoiding dropping off to the bottom right of that curve? That's the part of this that we're looking at now. So as I was kind of mentioning, we have these similar regions in the genome that make alignment difficult. This, back to the seeds explanation and the false positives, the odds of finding too many matches in the genome are so low if we had the random string assumption. But the fact that we have similar regions are what makes alignment take longer for us. So this is an example here. You see that it's colored by the type of base. So when we see a solid color column, that means all the strings are matching exactly. And when we see different colors in a column, that means some of the strings have differences from each other. And these are some strings that we found via clustering in chromosome 22. They're all unique. It's around 400 of them. However, if we find the consensus string for that group of substrings, and we find the average distance from each string in the cluster to that consensus, it's very low, only about 6 edits. So this is the phenomenon that's causing us to spend most of the cycles in the alignment process. So,
are those all from the same person?
So this is from the reference genome. And
almost always identical. Right? It's 1 part per 1,000 difference between people, so.
Yeah, so
and the reference genome came from a single person?
No, it's actually kind of a mix because different sequencing centers were working on it, so they all kind of submitted part of it, and it's kind of a stitched together. Yeah. So what we really want to be able to do is test that our read against the entire group of strings. However, that takes a really long time. So we have this trade-off then, which we've kind of looked at, and if we are too aggressive in lowering the max hits, then we're going to be paying the price in error. So just to illustrate this graphically, what we have in many cases is these reads that are matching against these similar regions. And so based on how we set max hits, we have some different problems. So if we take it too low, that means we won't try any of the locations that the read is going to match against because all of the seeds have too many hits. So we won't be able to align that one at all. If we take it too high, we'll try all of the locations, but then we'll spend an inordinate amount of time trying to align that read. So what happens in practice is because we have some middle of the road setting for max hits, what we end up doing is we test some of the locations where that read would match, but not all of them. And therefore, this is where most of our alignment errors are coming from. So, what happens is we test it against a few locations, we find one that works well, and we report that. However, there was actually a better one somewhere else that we didn't test. So this is the main thing that we're trying to address. And this is important definitely for the downstream processing. So we really want to be able to get this right. So what is our approach to fixing this? Well, first, we're working on pre-computing those similar regions in advance so that we can use that information quickly during the alignment process. Second, we're exploring an algorithm to, rather than you get the cluster and then you compare individually against each string in that cluster, we're working on a representation that kind of reuses the work in that comparison so that you can efficiently do that. So currently, this is a work in progress, but what we have so far is we have a parallelizable algorithm for detecting these similar regions using Spark, which that's a project that Matej's worked on with some folks at Berkeley and is a really good framework for cluster computing. And we also have a group edit distance algorithm that gets quite a good speed up over comparing against all the strings fully. So it's the way that we're detecting these similar regions is very simple, but I'll just quickly illustrate it graphically. So what we have is for each substring in the genome, we represent it as a node in the graph, and then we look at the other strings in the genome and we compute adjacencies. So this one only has 2 edits from our original substring, so we draw an edge. However, in some cases that have too many edits, we don't draw an edge. And then we, whenever we find that 2 clusters share a member that, or have members that map to each other well, we're going to merge those clusters. So we continue to do that. And eventually we end up with these disjoint clusters, and then each one of those would be the group that we want to compare a read against. And so of course this is expensive because we have this n-by-n adjacency matrix that we want to compute over the whole genome. So it takes too long to do it naively. So the way that we've worked on this is we've partitioned the this matrix so that each Spark task will be working on a separate piece. And what we do is we index this only the short part of the genome that this task is responsible for. And then what we do is we run our union find so that we get a set of clusters. And we do this for each of our tasks. So then now we want to be able to merge these clusters so that we get the actual set for the entire genome. And the way that we do this, remember, each cluster has a bunch of different strings in it. And whenever we find strings that match across clusters, we'll merge those. And we continue that for the whole batch, and then we get some final set of clusters over the entire genome. So a very simple idea. So
do you need to do this once per species?
Yeah, so definitely you would. So far,
you need Spark. Why not just do it and take a few weeks?
Yeah, I mean, I guess certainly you could do that. We've been focusing on only human. Yeah, we've been focusing on only human. You could certainly run this on whatever reference genomes that you wanted to work with. But so we've focused on getting a parallel implementation just because it's proved to be intractable to work on for in a single node.
Well, you only needed to run it once per species when it's done, when you're developing it. You run it once per species, so Crystal's own sanity.
Yeah, the exact length of the clusters might change. We don't know what the best way to really.
Yeah, and this is also something we're still kind of working on tuning the parameters too. So we'd like to have a shorter cycle for running it. So, but yeah, it's definitely this is something we pre-compute, so it's not going to affect alignment performance. OK, so then how do we use this information in SNAP once we've gotten it? So back to the case where we have a read that is going to match against a bunch of different places in the genome. What we do is we notice that one seed is matching against one of these flagged locations. So we kind of flag them in advance if they're belonging to a cluster. And then what we do is we just grab all the fellow cluster members of that location. And we just use that edit distance kind of aggregate algorithm I was mentioning to test against the entire group rather than testing against some fraction like the current version of SNAP does. So in this way, we can kind of get some of the best of both worlds in that we're saving time, but we're still able to compare against all the strings in that group. So we're able to avoid errors. And in fact, what we see is that once we've incorporated this into SNAP, if we compare against the standard form of SNAP, we get a big reduction in error. So this confirms the fact that really the errors that we were getting from the standard version of SNAP are caused by these similar regions. So, and as I'll kind of get into in the last part of the talk, these alignment errors can really be important downstream. So even though it's kind of a small fraction overall, it can lead us to drawing some bad conclusions if we have those errors. Okay, so what is the rest of the pipeline? So back in the beginning of the talk, Matej kind of talked about how the idea of reconstructing a genome is taking all these reads, aligning them. That's the part we've been talking about so far. And then, but the main goal is to know what that individual actually has for their genome at each position. So that's the process called variant calling. And in the kind of the simplest view, it's just this idea of taking a consensus of all the reads that map to a particular location and seeing what they indicate about the genome. But actually, this is a very difficult process, and we really are focusing on trying to improve the accuracy of this. So why is variant calling difficult? There are several factors. And first, there are some that are just inherent to the genome that make it difficult. So these aren't going to be going away. The other ones do have to do with technology, so they can potentially be targeted. But there are things that we have to cope with for now. So one, when we're actually taking the DNA out of the cells and producing the sample that's going to be sequenced by the machine, we have this process called PCR, polymerase chain reaction. And this is not an unbiased process for kind of amplifying the DNA so that we have enough to sequence. Again, we also have systematic problems that the sequencers make when they're producing the DNA reads. And as we've talked about, the alignment errors, that's also going to confound variant calling somewhat. Okay, so the heterozygosity. What this means is that for all of your chromosomes, you have pairs of chromosomes that are the same length, and you get one from your mother or one from your father. And so at each point on those paired chromosomes, if you have the same value on both of the strands of that chromosome, then this is called homozygosity. But if you have different values on the two, then this is called heterozygosity. And so why is that a problem? Well, when we get reads that map to that same location on the genome, they could tell you different things about what the person actually has at that site. And so it's not always straightforward to know, is this actual true heterozygosity, or is this just that we've gotten sequencing errors from a couple of the reads? So this is one of the challenges. Another challenge is, as Matej was kind of alluding to, we have something called structural variants, which are larger scale changes. So far we've been talking about mostly mutations that are one, maybe up to a few bases in length. But these are changes that can be arbitrarily large, even up into the megabases. So, and especially in the case of cancer, you really see a lot of these large scale changes. So what do they look like? Well, first you can have a deletion from the reference genome. And one way that you can detect this is that you have a bunch of reads that map normally, but then you have a segment of your reference genome where no reads map at all. And also, so Matej mentioned that you get these reads in pairs. Usually you know how far apart those pairs are. And sometimes you see that the distance between pairs is off from what it has to be given the parameters of the sequencer. So in this case, reads that actually come from a certain amount are looking like they're way too close together. So that's one large-scale problem. Another thing is you can have a sequence inserted. This can be from somewhere else in the genome, or it can be a completely novel sequence that you won't be able to detect at all. So in the case of a completely novel sequence being inserted, you'll have some reads from the patient's genome that can't be mapped anywhere in the reference genome because it's a novel sequence. Then also, you'll have some reads that came from close together, that should be close together in the reference genome, mapping way too far apart in the patient's genome. So, so these are also some of the things that make variant calling more difficult. Another is you can have.
How would you know where they mapped in the patient's genome to know that they were far apart in that mapping? Or is that through paired ends?
Yeah, you definitely always use the paired end. Okay, so yeah, that's a very valuable signal. So one of the trickier types of structural variants to look at is these duplications. So in this case, this is called a tandem duplication because you have a part of the sequence duplicated so it's adjacent to the original sequence. And what makes this difficult is that one thing is that you see too many reads mapping to that portion on the reference genome. But then you also have some reads that are kind of from the overlap of these two that are not going to be mappable because they're just going to look funny. So these are very difficult to detect, and they're still definitely an open problem for developing better tools there. We're looking into that. Another thing that makes variant calling challenging is that, as I was kind of alluding to, the process to converting the DNA to a bunch of short fragments that can be sampled during the sequencing is biased. And what are the biases? So one is the molecule length, and another is the composition of the molecule. So
just on the structural variants, are existing tools trying to define those, or are they mostly focusing on the SNPs?
Yeah, so there definitely has been a lot of work in finding structural variants, but the accuracy is still pretty bad. And also each tool is kind of like a point solution. So some are good at finding short insertions. Some are good at finding large deletions. Some can find more types of structural variants, but not very precisely. So it's still kind of like not clear how to do it for, in a way that is holistic and can find all the different structural variants. So yeah, that's why that problem is still very interesting. And especially because structural variants actually have a lot of interesting influence on cancer and other types of diseases.
I think I'm not, in a mutation, does it kind of look like heterozygosity, or does a mutation typically go to both chromosomes?
Yeah, both can happen. I mean, of course, usually you would have only one, but then in some cases you might have both of the genes knocked out on both of the chromosomes, right? So then.
Because I guess I don't know what causes mutations. I think of cosmic rays, which would probably only affect one chromosome. But
yeah, they can, but then that's kind of why cancer takes a long time to get, because it's just like accumulating the bad luck over like many, many years, right?
But also what happened is like in the human population, there were mutations long ago that now a lot of the population has. Like it's hard. There isn't really like one gold standard like human genome, and people have mistakes relative to it. There's just diversity.
Right, and so you can wind up inheriting two copies of a, right, if you look in the population tree, maybe there was some mutation that happened in chromosome 14, and one individual copying of that in the germline, and then it spread out the population. And you may actually, both of your parents may have gotten that through, the fact that your great-great-great-grandparents sometimes are the same as each other, right?
I'm Jewish, I know.
So it's mathematically necessary.
This doesn't be true for everybody, right? It's the old thing where you start counting back generations and you do 2 to that power and it's bigger than the population of the planet, and people get confused and say, what happened? Well, what happened is somebody is multiple times your grandparent, and that kind of thing can lead to getting the mutations in both copies, because you have a chromosome that came to you.
Okay, so yeah, so for sample prep, what makes this difficult is that there's biases toward the size and the motif. And what this looks like is, you start with your original full strand of DNA that you've replicated, and that you've gathered from the sample, and then you send it through some heat process that fragments the DNA. But then what we do is we apply PCR to amplify those fragments so that they can be sequenced throughout the sequencer. And you see that some fragments are preferentially amplified. So this means that, like I was showing you here, a higher coverage depth at that duplication. Well, sometimes a higher coverage depth can just be because of the way the sample was prepared. Another thing that's pretty subtle is strand bias. So I'll walk through this. This is from a visualization tool from some folks that created a lot of libraries for looking at genomic data. And what you see at the top, this TTTC, et cetera, is the reference genome at this position. This is in chromosome 20. And then what you see, these little dots and commas, these are reads. So the dots are reads that are mapping in the forward direction, and the commas are reads that are mapping in the reverse direction. Yeah, so yeah, I mean, we talked about paired end, and what happens in paired end is you read them in the reverse direction. So one gets read forward, and one gets read reverse.
Right, DNA is paired, right? You get the double helix thing that you always see pictures of, and in the double helix, the bases, there's bases that go together, like types of bases that go together, and they're directional. And the direction in one helix is one way, and one's the other. So the information in the two copies of the helix are exactly the same, but they're in opposite senses. And when the machine goes off and reads them, you can't tell which one you got. So one of the problems that comes in alignment and mapping that we didn't mention is that you have to check both senses, whether it's forward or reverse complement, and then this just tells you what sense the thing was mapped.
Thanks. So, yeah, so what we have here, the reads that you can see that are these commas are showing a bunch of T's. So where you see letters, the reads are reporting a difference between the individual and the reference genome. So, but you see that these T's are only showing up on the ones that are going in the reverse direction. And so this is a case where we don't want to trust the reads when they're telling us the difference from the reference genome, because they're only showing up in one direction and not the other. So this is just one and probably the simplest example of a strand of a sequencing systematic error that you can get. But definitely we have some other cases as well. So also this systematic errors in the sequencing can also show up based on the motif. So what we see here is so for one thing, we see more errors when the reference sequence is a T, and which we see with the red T there. The other thing is that we see more errors when the preceding two bases to the position are both Gs. So just because of the way the chemistry works in reading off the DNA in the sequencer, it tends to favor, or I guess it tends to have a harder time in these cases. So the fact that these errors aren't uniform makes this process more difficult. And a further thing is that variant calling is actually really difficult to evaluate as well, because we have very limited ground truth for this. And so one thing that you can do is produce samples of the genome with different sorts of technologies, and then compare what you get with the short reads to what you get with these other technologies. But there really aren't a lot of standard benchmarks that you can use to evaluate variant calling. And in fact, all of variant calling works thus far has kind of relied on ad hoc methods of evaluation. So one thing that we're working on is developing more standard benchmarks for our own development, just so we can know if we're actually improving based on the things that we're doing. So then another thing you can do, if you have the data that's really cool, is using this trio data. So what's a trio? That's two parents and their child. And the reason this is really valuable is because there's certain assumptions that you can make that the child's DNA has to be consistent with their parents' DNA. And the reason you can make this assumption is that the rate of novel mutations from the parents' DNA to their child is very low. So therefore, if you're seeing something strange, it's much more likely that it's a problem with the sequencing, the variant calling and whatnot, rather than actually a true novel mutation. So just in some of these examples, you see that the child's DNA is consistent with the parent's DNA because each of the child's sites on their gene is from one of their parents. But then, for example, these cases are invalid because their DNA doesn't match against one of their parents. So this is another really valuable signal when we have trio data that we can use for evaluating SNP calling. So this is still very preliminary, but one thing that we did, we wanted to check against the existing tools and get more of a clear benchmark on how they were performing, and also be able to try out some simple ideas and see if we could do as well or even better than the existing tools. So what did we do? We took some real data. This is, it's called the Yoruban trio. It's from some individuals from Africa. So we have two parents and a child. we looked at only one chromosome. So we aligned all the data to the reference. We took the reads out that mapped to chromosome 20, which is one of the smallest ones. We realigned it then once we got those reads that filtered onto chromosome 20. And then we did this, this SNP collar that we developed as a prototype. And we evaluated it based on those Mendelian conflict assumptions that I just mentioned. The other thing that we did is we looked at simulated data. So the simulated data, this is a simulator we've been working on in our group. And it's kind of meant to be more realistic than existing simulators because it uses some data with real SNPs that people have found in various sequencing projects. But we restricted the simulator to only include single substitutions. So it doesn't have any short indels or any structural variants. So in our thinking, the simulated case should have been pretty easy. And we did something similar. But of course, in the simulated case, we know what the variants are at each site. So we can actually get the accuracy there. So what are the preliminary results? Well, so we looked at two different pipelines. This is kind of a preliminary evaluation, but what we looked at is the Cassava pipeline, which is produced by Illumina, which is the main sequencing company that we've been talking about. And they called out about 128,000 SNPs, and they had 162 conflicts. So then we also looked at the GATK on the simulated data. And the GATK, it stands for Genome Analysis Toolkit. It comes from the Broad Institute in Massachusetts. And it's pretty much the state of the art for variant calling. This is the one that's most widely used by practitioners throughout the field. And what we found on the simulated data, even though it was very simple, is that there were still 66 false positives there in a pretty short amount of DNA just from chromosome 22. And it's also very slow. It took 4 hours to do this process. So what did we do? Well, on the trio, while calling about the same amount of SNPs, we got about half the conflict rate. And on the simulated data, we got an order of magnitude less of errors. And this is something that you can run in 2 minutes instead of 4 hours. So we think that we have some insights based on this that we should be able to further improve this to work on the full genome and also extend to other types of mutations besides plain SNPs. And so looking forward, our efforts over the next few months are focusing on kind of formalizing this feasibility study into a full-scale pipeline that takes us from DNA reads to a fully reconstructed genome. And so looking at the way that this is currently done, people have broken this up into individual stages and then kind of separately optimized each stage. And then in order to get the data from one stage to the next, they use a lot of thresholding. So whenever there's uncertainty, you have to just kind of cut it off, report one value, and then push the data on to let the next stage handle it. So kind of leveraging all the work that's been done, we're kind of hoping instead of having this kind of disjoint pipeline, we're working on producing this end-to-end system that will, rather than truncating information very early on in the process, we're working on propagating it through the entire process. So therefore, we're able to leverage that uncertainty in a better way than just throwing it away early. OK, so just to pop up a level, as Matej mentions, DNA sequencing is getting really realistic in terms of the affordability. It's improving in accuracy, and this is really going to have great impact on medicine in a variety of cases. And one of the things we're really excited about is about producing better treatments for cancer. And the cool thing for us is that it's actually involving a lot of computational challenges in addition to wet lab challenges. So we feel like we can participate because a lot of the existing tools lack in both speed and accuracy, and that's becoming a big problem if you really want to use this in a clinical setting. And based on our work, mostly with SNAP, but also our initial work with the rest of the pipeline, we have some evidence that we can help with both of those cases. And so if you're interested, you can check out our website. We've made this open source. We have it available for download. Actually, we presented it at a conference a few days ago, and so we've already kind of gotten a lot of interest, people trying it out. Yeah, so I don't know if there are any more questions.
Yeah, so I have a question about the kind of the end-to-end process. So you guys, we saw the error rates for alignment, and I'm curious if you've run any studies to see how that propagates down to variant calling.
Yeah, I mean, we've done some benchmarking, but it's been mostly just kind of running BWA, just since that's kind of the standard benchmarking, and just seeing how what our ultimate variant calling results are. But we haven't really done something where we kind of vary the parameters so that we get better or worse accuracy, and seeing how that gives us the variant calling. But yeah, I mean, definitely that's something that we're interested in looking at.
But even the comparison of, say, SNAP for versus BWA, do you see that the error rates, the difference in the error rates is actually something that helps with the variant calling process?
In the thing that was shown, there was at least one example of like a read that BWA mismap that we didn't, that caused it to call mutations. But this is a very it's a very anecdotal thing. We need to do the same thing on a whole genome, and we just haven't gotten very far with it.
Yeah, and part of the obstacle is just that GTK is so slow, right? We haven't even been able to run it on a whole genome because it's so expensive. And
at the conference we were at, I listened to the guy who runs the 1,000 Genomes Project talking about this, and the errors that have come out of BWA have caused them such problems that he was talking about giving up on remapping altogether and doing de novo assembly for all of the genomes because it was driving him bonkers. So that's some evidence that errors are a problem. We're hoping we can do better. And by building an integrated pipeline, you can correct for the errors later. Actually I don't know this. Does GATK do anything with unmapped reads?
No, not with unmapped
Our
reads.
plan is to use unmapped reads and to remap stuff later as you're looking at the, because there's lots of stuff you can tell is just wrong.
Yeah, I mean, other
sets of reads that you can't tell they're just wrong.
Yeah, so other tools do look at unmapped reads, but they're not integrated with GATK. So yeah, I mean, that's another obstacle, is just kind of making everything work together. And I mean, just in our benchmarking process, it was pretty painful to try some of these things out, so.
I was wondering about something I haven't wondered before, but after seeing the earlier part of the talk and the later part of the talk. So the approach is to take seeds from the reads and map them to the genome. And depending on the depth of coverage, you're going to have many more seeds to look at than the places in the genome to compare, right? But the complexity seems to be the same if you, I mean, theoretical complexity, not real complexity. If you go the other way around, if you actually take the seeds from the genome and try to find them in your reads. And again, you can do the same trick with stopping early, but now the stopping would be based on a different criterion, that you want to see a certain number because you know what the coverage is roughly, so you won't stop after you've matched a certain number of times. So has anybody thought about if that could be worked out to be almost as efficient as the other way around? Because then some of the structural variant and other issues would be perhaps easier after the fact. So you would pay a price a little bit. But I don't know. I haven't thought about it much because it just occurred to me now.
So we've thought about doing something similar to that for trying to do de novo assembly of structural variants. So the thought was you'd go through, get rid of the easy stuff, because most of it's easy. And most of the stuff maps unambiguously with few errors, and you can look at it, call it, and get rid of it. And then go back through the remaining reads and try to build them into contigs, which is, so you find overlaps. There's a couple different ways to do it, but essentially you find places they overlap, and you build long strings of DNA that they seem to represent, and you try to fit them in to places where you found breakpoints in the genome. So at that point, you would do that. Whether it makes sense to invert the process from the beginning, I'm not sure. I mean,
so the issue, the thing I didn't understand is it doesn't really help with structural variants, because it still tells you these reads look like they came from here, but it seems
from multiple places,
like ultimately you need to do this detective work at the end. Maybe it will help you with the reads that are split across both ends. Yes. But it's symmetric. Yeah. But some algorithms have done this. So, for example, some versions of BLAST do this because they just take the reads, say 1 million at a time, and they index those, and then they scan through the reference genome when it's too large for them. So, in a way, it's been done. But I'd have to think more about how it affects what you can do.
I haven't thought about it much, but it always, when you think about this problem, you always think about we're going to map the reads.
Yeah, yeah.
But I don't know. And in the end, if it's really alignment, then maybe in learning the process could be worked out. It's not going to be straightforward.
Yeah, I mean, there definitely have been some aligners that choose to index the reads instead of the reference. I mean, they're a little bit old, so I don't think we could really compare directly against their performance. But also, one aligner whose goal is to find actually all the matches for any read indexes both the reference and the reads, and then kind of walks through it that way. And then there's also been some work for detecting structural variants where you use assembly to reconstruct the patient's genome, and then you align the reads both to the reference and to that reconstructed genome. And then that's kind of a signal for how you can locate these structural variants.
Even though that may have been done in the past, just like the way BLAST was using seeds, and you're able to use that idea much better, because a lot of these things left, algorithmically, they're imperfect, so maybe.
Right. Yeah, that could be an interesting thing to incorporate.
Yeah, I mean, I think there'll be a subset of the reads for which it works very well. I mean, if you're dealing with reads that only have a couple of matches in the genome, the current process is probably the best, right? I mean, it really depends on how easy it is to find the match in the opposite strand. Right. But for reads which have a bunch of matches in the genome, the reverse approach might be better. So I think it's a really valid
point. Suppose you have a specific goal in mind for one particular patient. You don't need the entire sequence, but you want to know which of the 70 colon cancers he has. Can you make it holistic even beyond that and say, okay, this is the end goal. I want to update the whole pipeline to optimize for answering that one question I have.
I see, yeah. So one thing people do is they narrow their focus when they kind of gather the initial reads. They get it from only what's called the exome, which is the coding sequence or the genes that are in your genome, because that's actually only about 1% of the entire genome. You also have all this other stuff that orchestrates how, which genes get expressed, or could have just been randomly inserted by evolution. We don't really have a good sense for. So some people do just pull out that exome, sequence that, and then see how, if they have mutations where they think they might be. But the problem with doing that is that there could be a lot of other mutations that are related to that one that you are missing if you do that. So it's kind of like the looking under the light post approach, where, which is somewhat problematic when we still don't understand a lot of the other things that could be related. But yeah, in terms of how the pipeline might change, I mean, if you're focusing more on certain areas, then everything becomes a lot easier, because you've really narrowed down the uncertainty, so you know which parts of the genome that you're looking at. And then it's also going to make a lot of the stuff we were talking about with similar regions not as much of an issue. So